Okay, so here we are today. Next week will be our first lab pending any weather catastrophes. So make sure and go to the online syllabus and download or have open on your computer in lab the blood glucose lab handout. And that's the lab where you'd be pricking your finger with a small gauge needle about 3 times during the lab, okay? Try to avoid eating something really sugary or drinking something really sugary 30 minutes before lab if you can. If you have diabetes, ignore any of those things. Do what you need to do to manage your diabetes. But if you're otherwise healthy, just 30 minutes before lab, okay? All right, let's get this out of the way. So, do I have the PowerPoint open? Maybe not. Oh, I do. Okay, so I have updated the PowerPoint just this morning to provide a review slide. There we go. Try and make this as big as possible for you, and no recording. So before this, oh, backwards, we finished off, I was showing you glycogen metabolism, making glycogen, breaking it down, which is right here. And so if you click here, you get a blank flow diagram for practicing, and then here you get the key, and that's the key, no pun intended, for learning these pathways, because there's quite a few pathways in this chapter. So I put this review slide in to remind you, since it's been a very long weekend, what we covered on Friday. So we talked about metabolizing these 4 molecules. We did vocabulary word, lots of G words, so get your index cards. So we defined glycolysis, and then in making or breaking glycogen, making its glycogenesis, breaking its glycogenolysis. We talked about the enzymes involved in all of that process. So that we've covered. So today, we're going to start here, which is cellular respiration. You learned it in intro bio. Y'all hated it. I hated it too. But I've really simplified it. I need more tea. And just included what you need to know as future nurses, because is this relevant to everyday nursing? It actually really is. It's amazing how much intro bio or bio 105 is important in human physiology. It is. So we defined this before. Glycolysis is using glucose for cellular respiration. And let me add, see if my pen's awake. Oh, I don't like red. To make ATP, ultimately to make a whole bunch of ATP. That's the energy currency of our cells. That's what keeps the cells alive. Now, I'll go over this in a second, but here is what you may or may not remember from intro bio. 1 glucose molecule, and I made this diagram because I like it. It's very simple. And let me move this thing out of the way so it's not. Put it up there. You take 1 glucose molecule in your cells, because remember, insulin tells your cells to take up glucose. Here's what the cells are doing with it. The glucose goes through glycolysis. And what I've got at the end of that pathway, I've got the main product. And that will be, oh, sorry, my pen is acting as a selection. This is the end product of glycolysis. Off to the side, I've got side products. And that's how I'm introducing it to you. The main product is going to go further into other steps. The side products are, well, side products, and I'll explain. The only thing that's important is these H's will come into play in the last part of cellular respiration. So the main product I've put in bold, and the side products are here. So this is NADH2, and you take the 2 times the 2, that's 4 hydrogen ions. And you make a little bit of ATP, not enough for the cells to live, but they accumulate. Now, if your cells have plenty of oxygen present, they've got a good arterial blood supply, which is bringing things like glucose to your cells and oxygen. If oxygen is present, this process will then continue on other steps. And this is the ideal pathway for our cells, is to respire in the presence of oxygen. Our cells can make ATP without enough oxygen, but it's not going to allow the tissue to do it very long. Our skeletal muscles do it all the time. They can respire in the absence of oxygen. You've all experienced this. If you've ever climbed the 3 flights of stairs in Leatherman or McNair and you're feeling the burn in your quadriceps, right? You've burned up a lot of oxygen present in your skeletal muscles. They still need to make ATP to keep functioning, but by switching to anaerobic respiration called fermentation, they will produce lactic acid as a byproduct in addition to a measly amount of ATP. And that burn that you feel is your cells being forced to metabolize in the absence of oxygen. Our skeletal muscles can get away with it. And if you push them too far, they're going to give out. All right, they're going to fatigue and give out. If your brain or your heart itself, which are oxygen hogs, are forced to respire in the absence of oxygen, like they've got an interruption in blood flow, they can anaerobically respire. They'll only produce a little bit of ATP, not enough to keep the cells alive, and they will produce lactate. That's an emergency. And what happens when your heart has an interruption in blood supply? What happens to that person's heart? I mean, in a general sense, what happens when there's a blocked coronary artery to the heart? Real simple. Anyone blurt it out? Heart attack, myocardial infarction. Yeah, that part of the heart that's not getting the oxygen can die. All right. What's it called when someone has an interruption in blood supply, which interrupts glucose and oxygen supply to the brain? There's a term for that, begins with an S. Stroke. And is that person at risk of dying very soon if the intervention is not done? Yeah, because what's going to happen is the brain is going to respire in the absence of oxygen. It's only going to produce a little bit of ATP. Brain cells can't live on it, they'll die. The brain itself will produce lactate. All right. Now, is this relevant to nursing? Yeah. Okay. This pathway is. Okay. So I made another simple diagram because I like simple diagrams because I hate complex ones. It doesn't have to be that complicated. So glycolysis has happened. And let me correct this. We have now got, go back. The main product is 2-pyruvate. The pyruvate can then go on. So glycolysis, and more specifically, once you make pyruvate, which is an acid, it can go one of 2 pathways. If there's oxygen present, it can go on 3 more steps and it can make a bunch of ATP. Your cells are all happy, homeostasis is maintained, and you're golden. If there's no oxygen, pyruvate goes through anaerobic respiration, and the products are 2 ATP, not enough to keep any cells alive, and lactic acid, just known as lactate. If it's lactic acid, what do you think it does to your blood pH? Drops it. It causes acidosis. I think we explained last time why acidosis is bad. Okay, so 2 bad things happening. You're spilling a bunch of acid into your bloodstream, and you're not producing enough ATP in those cells, and that's going to be an emergency in your heart and your brain. Not so much your skeletal muscles, they do it on the daily. So if oxygen is present, that pyruvate goes through these steps. It gets processed. I'll show you that. Then the Kreb cycle. Does that sound familiar from Introbio? Horrible, long cycle with dozens of acids. And then the ETC, I'll explain later, electron transport chain. That probably sounds familiar too. And then one glucose molecule, which is converted by glycolysis into pyruvate, can make a bunch of ATP in our cellular party. Having a great time, no problem. All right. Next week, and not next week, but the week after, we're going to be doing a lab where we force cells to metabolize in the absence of oxygen. And we can't measure the lactic acid, but we can measure something else as a byproduct. We're going to use yeast cells because we don't have human cells. That would be very expensive. But we're going to make yeast cells try and metabolize a sugar in the absence of oxygen, and it'll produce CO2 waste, much like our cells do. And we can measure that CO2 and look at metabolism. So from here, nursing application, ischemia, that's how this is pronounced, serum lactate, and heart attack or myocardial infarction. So ischemia simply means a loss of blood supply. Any tissue can experience that. So without arterial blood and the oxygen that's being carried on our hemoglobin, resorts to anaerobic respiration, and then the heart muscle can die, not the whole thing, just what is downstream of the blockage. The pain that you feel, and some people don't feel this. Some people are walking around having a heart attack, they didn't even know it. You'll see when you're doing your rotations in the cloud. All right, they'll come in for something else and you realize, maybe you do an EKG on them for something else. Oh my God, you're having a heart incident. Really, I feel fine. You're not fine. But sometimes people have symptoms, and that is pain in the chest and pain radiating through their left arm. Women tend to show heart attacks differently. They'll get pain in their jaw and in their back and things like that. But a lot of men show that angina pectoris. Pectoris means chest. The pain is because the lactic acid is building up, like the pain you get in your skeletal muscles when you push them too far. The lactate is causing the pain. All right. So the loss of blood flow to the heart can lead to angina pectoris, which is the pain. It can be a prelude to a heart attack or myocardial infarction. So if someone calls up to the hospital and they say, oh, my husband or I have been having this pain in my chest, it's going down my arm, come in. Maybe it's nothing. You think I take to come in? What if I try to come in? Because the longer the heart goes without a blood supply, the more likely heart tissue is going to die, and it's a permanent problem versus if you catch it early and help them. Now, don't memorize this, okay? But there is a normal range for serum lactate. And when it's high, and you can measure it in a blood panel, it's called hyperlactatemia. So hyper means high, obviously. And then the word lactate is in it. And then you overlay emia, which means in the blood. So literally it means high lactate in the blood. And so if someone comes in and you suspect they're having a heart attack, they're going to get an EKG. They're going to get blood drawn. That blood is going to go to pathology and it's going to get a blood panel. You're going to be looking for lots of things like heart muscle. You're going to be looking for serum lactate. I'll show you that in a second. And you'll be looking for other biomarkers of the heart's experience to heart attack. Now, so if you click here, when you're online looking at this PDF, but I'll pull up right here, ischemia refers to inadequate blood flow to any organ, inadequate blood flow to the heart, or myocardial ischemia can occur if a coronary artery is blocked, all right? People with myocardial ischemia often experience angina pectoris, pain in the chest and left arm, but not all people having a heart attack feel that. The pain is associated with increased lactate or lactic acid produced by the heart muscle, which is being forced to respire in the absence of oxygen because there's a blockage, all right? The cells, if not caught early, can die, and that's permanent. Once heart cells are dead, that's permanent heart damage. And then you can do things like vasodilate the arteries. Don't memorize these drugs. But there's lots of things you can do to help that patient in the meantime while you're trying to figure out the problem. Obviously, if they've got a blockage, they've probably got to go to surgery or get on medications which can help dissolve that clot or reduce how much cholesterol plaques build up in their arteries. Big 3 in the PD: heart disease, hypertension, and diabetes. All right, and this falls under heart disease. All right, now, nope, don't want that. So back to this. So don't memorize this, just read it if you want. But serum lactate as a marker of acute myocardial infarction. So because this is relatively new, normally they're looking at CPK values to determine if someone's had a heart attack in addition with a EKG, but it takes a while for the CPK enzymes in the blood to rise, whereas serum lactate rises very quickly in the blood. And so it's being used as an indicator that someone's had a heart attack. So 5 million patients visit the emergency departments annually with chest pain. Fewer than 15% are actually having a heart attack. Some people, they feel this pain and they should always go in. They come in, they do the EKG, they do the blood panel, they look for lactate, CPK enzymes, other things, and they say, there's no indication that you're having a heart attack, but do you have acid reflux? Oh yeah, I've had it since my 20s. You know what acid reflux is? Gastroesophageal reflux. And some people, and there's a genetic predisposition, the cardiac sphincter to the stomach, where the esophagus brings food and liquid, that sphincter is not working like it should. And if someone's producing more acid than they should be in their stomach, that acid can back up through that sphincter and get into the esophagus. The stomach has lots and lots of mucus to protect it from acid, but the esophagus doesn't have enough to protect it. That acid is going to start causing an ulcer in the esophagus, and left untreated can actually perforate, cause a hole. That causes pain, and you feel it in your chest, and you feel like you're having a heart attack because the esophagus goes behind the heart. And you're feeling pain in the esophagus, you think, well, that's where my heart is. Oh my God, I'm having a heart attack. And so sometimes it can look like they're having a heart attack, but they're actually not. But anyway, baseline vital signs, EKG readings can be unreliable. Rapid assays of CPK or CK subforms, and I'll talk about that, may not rise for 12 hours, but lactate will rise before that. And then you can start to find out what's going on. As soon as you know what's going on with the patient, you can get on to treating them. All right. So diagnosis, quick diagnosis is good. So I'm not going to go through this whole article because you can look at it yourself. But now, is cell metabolism important to everyday nursing? Yes. And some of the quick tests that can help save a patient, diagnose them early and get them onto treatment? Yes. Okay. So aerobic respiration, if oxygen is present, no blockages. After glycolysis, you'll have pyruvate. Pyruvate goes on 3 additional steps. And I'll keep doing the main product and side products. And I'll call out the main product in a bright, bold color. So pyruvate gets processed. It's called pyruvate conversion. I don't know which way you learned it in intro bio, so I'll give you both phrases. So in that, here's the equation. 2 pyruvate enter. The main product is in bright red. The side products are in blue. So I'll put here, main product, side products. Oh, look, CO2 is a product of cellular metabolism. It is. All right, Acetyl CoA goes on to the Kreb cycle, known as the citric acid cycle. What's going to happen there is 2 acetyl CoA enter. The main products are the hydrogen ions. In fact, nothing is wasted. The hydrogen ions in the previous step will also be used, but hydrogen ions are the main product. They go on into the electron transport chain. I just abbreviate it, give it an acronym. Electron. You had to take basic inorganic chemistry to even be in this class. So hydrogen ions. How many protons does it have? 1. All right. It has no neutrons, but what it also has is, I'll circle it, 1 electron. Check the periodic table if you doubt me. Okay. Google it. Electron transport chain. The hydrogen ions that are produced in previous steps are going to be shuttled along the mitochondria of your cells. Oxygen that you breathe in is going to help in the process of making ATP. So this is the last step in aerobic respiration. All the NADs and FADs, and I'll show you a big flow diagram, enter. What they do is they combine with the oxygen you breathe in, H2O. So these hydrogen ions are reduced in the process. That's why at the end of it, the side products here, you've got NAD pluses and FAD pluses. That means the electrons have one hydrogen, or the hydrogen ions and their electrons have been used. And then you get metabolic water. So where do those hydrogen ions go? Bingo, combined with oxygen, metabolic water. How many people knew that from intro bio? Probably none of you, because they taught it real boring, and I know this. But the main product is all that ATP. That's a lot. Your cells are going to live on that. They're going to maintain homeostasis, and everything is good. So that's the value, okay, of the oxygen. It's going to interact here. Got that? That's as complicated. I'm not going to make it more complicated than that. That's it. Breathe a sigh of relief. No one wants to study all those pathways. Too much. So from here, I made this diagram. I like it. It's a flow diagram. Yes, it is. And do I have a blank one on my syllabus? Yes. But let's revisit what we've just done. Glucose. Insulin is in your bloodstream. Glucose will be taken up by your cells, lowering your blood glucose because now it's in your cells. The cells then take glucose and they go through glycolysis to make pyruvate. That's the main product. Off to the side, some NADHs and some ATP, just a little bit. Now the pyruvate, if oxygen is present, goes through pyruvate processing. The main product is acetyl CoA. Side products, oh, more NADHs, more hydrogen ions, and some CO2. The acetyl CoA goes into the Kreb cycle, and yes, I know each molecule of acetyl CoA spins it once, so it spins twice, but way too much information. You make a whole bunch of NADH2s, all right? That's basically 12 hydrogen ions there. And this is the first time that FAD makes its appearance. And there's only 2 of those. You make some more CO2 and a little bit more and ATP. Now, all the H's from here, and from here go into the electron transport chain. And at the end of that, the hydrogen ions are gone. They've combined with oxygen to make metabolic water, H2O, and a bunch of ATP. So what happens here is electron transport chain, let's say, just makes 30 ATP. How do you get this? Well, you add to the 30, the 2 ATP here, and the other 2 ATP here, that's 4. So the minimum you can make is 34 ATP. Sometimes you produce more, but that's what's going to keep your cells alive. Here's another way to remember this. When I ask you in a future exam, what is the main product of glycolysis? What's the main product of pyruvate processing? What's the main product of the Krebs cycle? Okay, and basically the main product of electron transport chain is a lot of ATP. I'm just going to ask for the main product. And so that's what you're going to be studying. Because what goes in, I could ask, and what comes out. This goes in, that comes out. This goes in, these come out. These go in, that comes out. That's how I'm telling you exactly how I'm going to ask it on an exam. Now, in terms of filling out the blank flow diagram, you'll be filling out the NADHs and FADHs. Well, the N, think more numerous H's, because there's going to be a lot of them. There's 2 here, 6 there. You see a lot of NADHs, but you only see FADH here. And so when I think of the F, I think fewer H's, because you only see 2 in the in the Krebs cycle. So these are the things that help me remember. Now, of course, no oxygen means lactic acid and just a little bit of ATP, not enough to keep any tissue going for very long. So this is it. It's not going to be complicated like in a textbook. If you needed to know more, you go to a textbook like any reasonable human being, because no one can memorize that shit. They can't. So here's what's happening with those hydrogen ions. And if you click here, when you're looking at this online, you can see this GIF, because the PDF, it won't be active. But the hydrogen ions are shuttled along your mitochondria. They cause production of a lot of ATP. Here's your mitochondria. It's totally intro bio stuff, all right? And I think that they're going to blow it up good. Okay, so the H's shuttle across. They're going to be used to make ATP. Don't worry about that. And then water is created when oxygen that you breathe in binds with those hydrogen ions as they're used up. So that's what's going on in the electron transport chain. Now, here's something I don't know if you'll ever see. Oh, here's the blanks. So I got asked, why do we need to breathe oxygen when I was a graduate student? And I was like, because you need it to survive. And we're like, it's more complex than that. What is oxygen actually doing? So I bet if you ask a nursing faculty member, why do we need to breathe oxygen? They're going to think of the simplistic answer. They might not actually have taken it down to a cellular level. Now, some people are really smart. We need to breathe oxygen because oxygen is the final electron acceptor, and the electrons are on the hydrogen ions in the electron transport chain in making ATP. The hydrogen ions are shuttled to make ATP, and then it binds with oxygen to make metabolic water, and that's why we need to breathe oxygen. So that is the answer. So if you click this when you're online, you get to the blank diagram, and if you click here, there'll be a key. It's also just on the syllabus. So if you want to practice this, you already know what I'm going to ask you on an exam. What goes in, what comes out. but I might ask you in the electron transport chain, the main product is lots of ATP. I'll ask you in a multiple choice question, how many NAD with the plus sign and FAD with the plus sign are there? All right, and then you got to remember, it's always going to be a higher number of NADs because N is more numerous, and there's only going to be 2 FADs because F fewer. So now you know exactly how I'm going to ask that. So I'll always tell you, I actually tell you exactly what I'm going to ask on the exam, and then you just got to study it. So cyanide, I doubt that you will see that in a hospital. You might. It's always portrayed in movies. The villain gets caught and they have a cyanide capsule that they break in their mouth and they die, and you would die pretty quickly. Here's what cyanide does. It's a very quick death. You got to go. This is a great way to go. It interferes with the electron transport chain. So what it does, make this a little bit bigger. Fast acting, it sure is. Cyanide, the coma, ultimately death, because your brain is not going to be able to make enough ATP, and your brain goes and then you follow. It blocks cyanide, the transfer of electrons to oxygen, the shuttling of hydrogen ions, and then binding with oxygen. You can't make that ATP that you need in the electron transport chain. It cuts off the electron transport chain. That 30 ATP that would be produced can't be produced. All you've got now is 2 ATP from glycolysis and 2 from pyruvate conversion. Actually, it's 2 from Krebs cycle, sorry. 4 ATP is not enough to keep your cells alive. So that's how cyanide kills you. It cuts that last step, which produces the most ATP. Okay. So now all this is a concept where cell respiration occurs. This is glucose in your bloodstream. This is a blood capillary. So glucose is in your blood. This is a cell membrane. So this right here is a cell from here on down. This is the cell cytoplasm, which is the fluid component of your cells. And this is in your cell mitochondria. So there's a line of separation here. So once the glucose comes in, it goes through glycolysis. You make pyruvic acid or pyruvate. It then can be anaerobically respired in skeletal muscle to make a little bit of ATP and lactic acid. So if this was a skeletal muscle cell, it's doing that there. Now, if there's oxygen present, the pyruvate, I'll just say, goes on to, I'll put right here, pyruvate conversion, because it's missing, which will make the Acetly CoA, which then enters the Krebs cycle, produces a lot of hydrogen ions that go through the electron transport chain, and you get a lot of ATP. So that's happening in the mitochondria, like that little GIF I showed you. All right, so glycolysis happens in the cytoplasm. All the other things happen in mitochondria. So that's just a concept. Okay, how are we doing? How are we holding on? Because this is a tough thing to, after a long weekend, to come back and talk about this crap. But like I said, I only try and include the parts that are going to be relevant to you as nursing students. All right, so now what we've covered is all that, believe it or not. It gets a little better from here. That was the tough stuff, all right? How glucose is used to make ATP. That was the hard part of this chapter. From here, it gets more reasonable. So we've talked about metabolism of carbohydrates, which is glucose and glycogen. We're done with that, yay. But we'll come back to it in a way. But now we're going to move on to metabolizing lactic acid. We need to metabolize that because our skeletal muscles are producing it all the time. If you're walking and moving around, your skeletal muscles are producing lactic acid. That's going to drop your blood pH and cause acidosis. You need to get those acids out of your bloodstream so you don't have a problem with that. And so lactic acid metabolism, I can't remember if I included, oh, I had a vocabulary word here. Let me move this for a sec, so it's not blocking it. So your body is an amazing machine. You already know this, and its metabolism is nothing short of miraculous. Your body can make glucose from non-carbohydrate sources. If you break down glycogen into glucose, that's still carbohydrate metabolism because glycogen in your liver, skeletal muscles, is a carbohydrate. What happens? How do people on ketosis make it? They've used up their carbohydrates. all right? And in their daily diet, they're depriving themselves of it. How can you make ATP if you don't have glucose? You can take fat stores, are here, and make glucose out of it. You can take lactic acid and make glucose out of it. You can take protein and make glucose out of it. This is the chemistry part, which there will be any chemical equations for you, but that's called gluco. Gluco means glucose. Neo, which means novel or new. And what does genesis mean? Create or make. So it literally means the making of glucose from a novel source, from a non-carbohydrate source. That's gluconeogenesis. That's why people, when they go through ketosis, they're trying to burn body fat, right? And the body fat, where's it going? The lipids are going to be mobilized in your bloodstream, and your cells can use it to turn it into glucose and make ATP. Lactic acid happens, now here I can't remember if I have the definition or not. Yeah, I did. Lactic acid is produced by your skeletal muscles, or in an emergency situation, your heart or brain if it's not getting oxygen. All right. So lipogenesis, or making lipids, when you have excess glucose in your bloodstream and there's insulin in your bloodstream, insulin tells your tissues to take in glucose. They take in that glucose and do the whole long process I just explained to you. Glycolysis, pyruvate conversion, Kreb cycle, and so on. Now, above and beyond what your cells need, your fat tissue, your fat cells, and your liver take in extra glucose above what they need, and they turn it into glycogen. Okay, so that those fats get converted into glycogen, and your liver can store fat as well. I'm blowing your mind, like, how the hell is that happening? I have a simple diagram that's going to show it. But your liver stores fat, and that fat can be used for cells to make glucose. What happens when your blood glucose is too low? Sensor, integrating center, and effector is what? It's about right here. Your pancreas. Blood glucose is too low. What's the hormone the pancreas secretes? Glucagon, because glucose is gone. Okay? Glucagon can go to the liver and tell it to break down its glycogen stores. Here's something else that will happen. The liver only has so much glycogen stores. When it runs out of that, and that can happen if someone's not eating, or they're skipping meals, they're sick, or they're anorexic or whatever. When the liver has blown through its glycogen stores, it's going to start using its fat stores and start releasing fats into the bloodstream. So what is that fat that people can get a urine test strip for if they're trying to burn body fat? What's that called? Ketones, ketones. And when there's a lot of ketones in your bloodstream, that's ketosis. Your liver will also release fatty acids into the bloodstream. Your cells can take ketones and fatty acids and use them to make ATP. That's going to be lipolysis. Okay. When you lack carbohydrates and say the liver's blown through its glycogen stores, your body turns to fat next. The liver will release a bunch of fat, ketones, and fatty acids into the bloodstream. Your cells can use that to make ATP. Fatty acids and ketones, what does it do to your blood pH? Drops it. So yeah, you can live on your fat stores, but your body's not going to like it long term. There's going to be problems. Now, if someone's in ketosis because they have this going on, and this is what I've heard from a physician, I'll be honest with you, I'm ashamed, but me and my husband, I think this was pre-COVID or start of it, and COVID weight because you're not active and whatever. You don't want to go to a gym because COVID. And so we were like gaining some weight and wanted to lose some weight. So we started avoiding carbohydrates, reducing them in our diet, and having things like meats, vegetables, maybe a little bit of fruit, stuff like that. But pastas, breads, no. We got into ketosis, yay, right? My husband went to his physician and got a health screening done. And actually I did too, because the university does it. And guess what was high in our blood panel? Fatty acids and ketones. Now, and triglycerides too. Our livers were dumping fats into the bloodstream to provide what the cells needed to make ATP. Now, cardiovascular disease. What's a problem? What's a leading cause? There's many things of cardiac disease. High blood fatty acids, high triglycerides. There's a normal range for them. So is ketosis and the whole thing that goes with that good for you? You might burn some fat, but you might start laying down some cholesterol plaques in your arteries, leading to a blockage. It's not healthy, okay? I guarantee you, someone who's going through ketosis, especially if you're doing it as a lifestyle, their blood triglycerides and ketones and other things, fatty acids, are going to be high. And now they might be burning some fat, but you're heading into cardiovascular disease. Not good, all right? So we stop doing that crap, all right? Okay, I know I digress, but this is how it's relevant to you guys. Now, amino acid metabolism occurs when you consume protein, you metabolize those proteins. You take whatever proteins you're consuming and turn them into human proteins. And with you, or with heavy skeletal muscle use, because you can cause a little bit of damage to your muscle that has to be repaired, or during starvation. Think of an anorexic or person who is so sick they're not eating. They blow through their carbohydrate stores fast within a day. They start blowing through the lipid stores. This can take a while, okay? Like weeks. Eventually, when they run out of the fat stores, the fat that you have under your skin and in your liver, they start moving on to your skeletal muscle, breaking down those proteins. And you start to see people's muscles wasting away, and that's getting close to end stage, all right? So this metabolism stuff is relevant to nursing. I'm keeping track of the time. Okay, lactic acid. You produce it every day, but your blood pH stays pretty constant if you're healthy because of your liver and the Cori cycle. So the Cori cycle recycles lactic acid. That's how I like to remember it. The Cori cycle, which your liver does, takes that lactic acid out of the blood so your blood pH doesn't drop, and then it turns into something the body can use. Nothing wasted. So the liver can recycle lactic acid into free glucose that the liver can spill into the bloodstream if your blood glucose is low. It can actually take that lactic acid, turn it into glucose, and then store it as glycogen for future needs, okay? It reverses metabolic acidosis because it gets rid of the acidic lactic acid. So this is happening in the daily for you. Your liver is doing this for you. And if someone has liver disease, they're going to have all kinds of problems. They're not going to be able to manage their blood glucose well, and they're not going to be able to recycle that lactic acid. The Cori cycle can't happen. So this is just a diagram. Your skeletal muscles are the main producers of lactic acid every day. Do not memorize this, but lactic acid is produced from skeletal muscle activity. Okay, so you've got lactic acid that goes into the bloodstream. Left there, your blood pH will drop and you'll have a whole bunch of metabolic problems. So what the liver does is it takes that lactic acid and it can turn it into pyruvate. That's just known as pyruvate. Okay. Once you've got pyruvate, you can go anywhere. You can take that pyruvic acid or pyruvate, convert it into acetyl CoA. The acetyl CoA can go into the Kreb cycle, produce a lot of hydrogen ions, and the hydrogen ions can go through the electron transport chain and make ATP. The liver can take what would be a bad product in the bloodstream and use it to make ATP. Okay. Now, the other thing is, let me review something with you. This is the first part of cellular respiration. Okay, and a side product is 2 ATP and 2 NADH2. Okay, this process right here was glycolysis. I want to give you an interesting concept here. We covered this way back. Glucose goes through glycolysis to make pyruvate, which can then, if there's oxygen present, do the other 3 steps. Okay, we've got pyruvate from a waste product that's dangerous in the bloodstream. Can pyruvate not only go this direction, can it go backwards through glycolysis? Yes. When I have the arrow It can go either direction. That's basically inorganic chemistry. Have you ever seen the bidirectional arrow? It looks like this, something like that. If it goes in one way, it can go backwards. That's what your body does when it's metabolizing things. So the pyruvate goes backwards through glycolysis and makes that intermediate glucose 6-phosphate. That was when we were talking about glycogen metabolism. You've now gone backwards through glycolysis to make glucose. That's happening all the time, all right? Now, what the liver can do, it can take the glucose 6-phosphate and make glycogen. What's the vocabulary word for when when the liver takes glucose molecules and makes glycogen out of it? Hint, hint, it's going to end in genesis. Glycogenesis, glycogenesis. This, going from this to this, is glycogenesis. The enzyme that synthesizes glycogen from glucose is glycogen synthase, the enzyme ASE that synthesizes glycogen. So see how I keep building this whole thing? I started with glucose metabolism, glucagon and insulin, and then I built up glycogen metabolism with the enzymes. And now I'm talking about metabolism of a completely different molecule, and those chemical pathways are all active. We've got stored glycogen now from a waste product. If the liver sensed glucagon in the bloodstream, it would take the glycogen, break it down into glucose 6-phosphate. The enzyme for that is glycogen phosphorylase. And here's something I'm going to do. I'm going to circle the L. I'm going to come back to that. But that only does the first step, breaking glycogen into this intermediate glucose. This cannot enter the bloodstream. The liver has a special enzyme called glucose 6-phosphatase that breaks this down into this and spills it into the blood. That's what glucagon is doing. I'm going to circle the T. These are little tricks of the trade, okay? If I have in a future exam, which enzyme that is found specifically in the liver, not skeletal muscle, can actually convert glucose 6-phosphate to free glucose that can enter the blood? Well, there's lots of enzymes, okay? There's glycogen synthase, there's glycogen phosphorylase, there's glucose 6-phosphatase. Here's two ways to remember this. What comes first in the alphabet, L or T? L. So that enzyme comes first in the breakdown of glycogen. So that's breaking down glycogen. Another way to look at it is the word, the thing that it's breaking down is in front. This enzyme is breaking down glycogen because glycogen comes before the enzyme. T comes later in the alphabet. That's the last enzyme in the process. Here's another thing. What is it breaking down? Glucose 6-phosphate. It's in front of the enzyme. It's in front of the ASE. Two ways to remember this for a multiple choice question. And if you remember those things, L comes before T. And the word, the product that's being broken down is in front of the enzyme. All right, and you won't go wrong. Because I know there's a lot of G words that you're going to be memorizing this particular chapter. So these are things that can help you remember the difference between them. Have I still got you? You alive still? We're getting there. We won't get it all done today, but we're getting a big chunk of it done. So again, I just introduced this vocabulary word. Yes, it's another G word. What is the term for when a non-carbohydrate molecule, in this case, lactic acid, are turned into glucose? It's making glucose from a novel source. So genesis means create. Oops, go away. So that's part of it. The neo is the novel. And what are you making? Glucose. Oops, sorry, CO. Making glucose from a novel source. Metabolism of lactic acid, you make glucose from it. You're making carbohydrate from a non-carbohydrate source, gluconeogenesis. It's enough G words, isn't it? I think there's some more in the chapter still. So this chapter is heavy on vocabulary, lots of G words, and there's a few pathways. All right. So, and we're going to end soon here because I'm looking at the time. Let's say you have insulin in your bloodstream, which happens all the time. Your cells take up glucose. I now told you that your liver and your fat cells can take that glucose and make it into glycogen. They can also take that extra glucose and turn it into fat. So we're going to do this now, lipid metabolism. So you can either make lipids, which is lipogenesis, and I don't know if I have the vocabulary words. No, I don't. Lipo is fat and genesis is lipids. So you make lipids. Okay. If you have above and beyond the glucose that you need in your body will make fat. And if you need to make energy and you don't have carbohydrates, you break down fats. Okay. So lipo and lysis. Lysis means to break apart. If you lyse something, it breaks apart. So I'm going to first talk about lipogenesis. You consume plenty of glucose. Your body's like, what do I do with it? I don't want to waste it. It's valuable. Let's turn it into fat and store it in case we go through some lean times. Like in the past, we evolved to store fat because you never know when your next meal was going to come, and it can help you survive for a couple of weeks until food comes along. All right. So making lipids. If insulin's in your bloodstream, your cells will take in glucose, make a bunch of yummy ATP. But if you get above and beyond what you need, fat cells turn the glucose into fats. All right. So triglycerides are stored fat. I'll say stored white, sorry about that. Stored white fat. They're called triglycerides. That's why the triglycerides are on every single blood panel. How much fat you got circulating in your body, all right? Because it could be an indicator of cardiovascular disease. So the extra glucose, what your cells don't use to make ATP, fat cells in the liver take that glucose. Oh, and look, they produce pyruvate because glucose enters glycolysis and produces pyruvate. That pyruvate, if the cells needed an ATP, you know what will happen from here. But let's say they've got all the ATP they need, they're happy. The pyruvate goes through conversion to make acetyl CoA. We know that process, pyruvate conversion. The acetyl CoA can be used by fat cells in the liver to make fat. So here's what happens. Acetyl CoA is used by the liver to make cholesterol. So cholesterol, you measure cholesterol in the bloodstream in a blood panel, right? If you suspect your patient has possible heart disease, you want to know if there's cholesterol plaques in their bloodstream. You can actually measure cholesterol in the blood as one of the things you look at to see if someone's got heart disease. The acetyl CoA can be used to make ketones. Ketones can be used for energy. Ketone is a fat. All of these are fats. But here, to make fats. Cholesterol is a fat. It makes up your cell membrane. It makes up steroid hormones. And if cholesterol is too high in your blood, it can clog up your arteries. Ketones are a fat that can be used for energy. And then fatty acids. Fatty acids get converted into stored fat. And we'll pick this up if we have class on Friday, okay? That is the fat that we have under our skin, around our heart. That's stored fat that we can use if we need it. All right, so hopefully I'll see you Friday, but if not, I'll come up with a lecture to finish this chapter. On Monday, we're going to be doing this next part of this chapter, which the PowerPoint has already been posted online. All right. Okay, stay warm out there. Oh, shoot. I think I closed the internet and I lost my, oh, bummer.